Hello, everyone, and good afternoon. Thank you again for joining our Fireside Chat. This is the second of three. We are coming to you from home offices and kitchen tables, sufficiently physical distance. And today we're going to talk about SARS-CoV-2, COVID-19, and in particular, we're going to focus on the immune response to this novel virus. I'm joined today by four panelists, all of which are card-carrying immunologists from the University of Georgia with various specialties to drill down into the abnormal immune responses that we're going to get into today and talk to you a little bit about their research programs and other research programs at the university that are contributing to knowledge dissemination for this virus. The panelists are Karen Norris, Balazs Rada, Kim Klanowski, and we're joined again by Chet Joyner, if you all joined us last week. So again, I'm going to put a plug in for the importance of academic research. These investigators that you're going to hear today and others from the university, they are knowledge leaders and they contribute the foundation of information for this particular virus and other viruses so that others in biotech and pharma can use the base information, the underlying mechanisms, targets in order to develop diagnostics and drugs. And I'm confident in saying that any drug, vaccine, or diagnostic that you take was born at an academic university, because without the underlying information that your faculty and researchers are disseminating, you wouldn't be able to get to a drug or a vaccine. So you all are going to hear kind of the workhorse research at the university these days, which is very strong in infectious diseases and immunology. And so with that, I'm going to get started, and I'll introduce you to Karen Norris, who's going to talk at a very high level about the various symptoms and why certain people are experiencing more serious symptoms than other folks in the population. And from there, we're actually going to drill down into these adverse events that are going on that are really mediated by your own immune system for various reasons. And so Karen is a Georgia Research Alliance Eminent Scholar. She's the Charles H. Wheatley Chair in Immunology and Translational Biomedical Research, and she's a professor in the Department of Infectious Diseases. Her lab conducts research in immunology and infectious diseases with an emphasis on vaccine and immunotherapeutic development. And that emphasis is directed towards respiratory infections within individuals that are immune compromised, that are advanced in age, that have underlying inflammatory diseases and or metabolic diseases. And so, Karen, I'll turn it over to you to get our chat started.
Thank you, Michelle, and good afternoon to everyone attending. I'm going to talk, as Michelle said, just very briefly so that we can get into the heart of the immunology, which is the most interesting part. But I just wanted to sketch out in one or two slides what we know at this point, 3 or 4 months into this pandemic, about this infection. And that is informing how we can treat these individuals that have serious complications, and also it informs how we are developing the vaccine that is going to become so critical. So I think everyone is aware or should become aware that this is a virus that none of us have seen before, and so our immune system has to learn immediately, as soon as we come across this virus, we don't have any memory from previous infections with this type of virus. And so most of us, because our immune system is so elaborate, and my colleagues are going to talk about the components of that, most of us do well and our immune system works properly. And so most people who encounter the virus do pretty well with it. The concern is the percent of the population that we're seeing now that have very serious consequences. And so some of those symptoms are shown on the right hand of the slide. So most people that come across this virus will develop some mild common symptoms. Some people develop no symptoms at all. But the dry cough and the sore throat, those and fever all seem to be pretty common. What has become more serious, and we've learned as things have gone on, is there are manifestations and complications of this virus infection in some individuals that really target organ systems from head to toe. And so there are heart symptoms, cardiovascular symptoms. There are central nervous system. So there's evidence of headache and kind of curious symptoms of a loss of taste and a loss of smell. So it tells us that the virus is affecting symptoms outside of the lung, which is where most of the clinical symptoms start and where the infection starts. What has become of grave concern is that percentage of the population that are sick enough to require hospitalization, and then those patients that are hospitalized that even require intensive care. What we've learned, and I think everybody is aware, is that it's individuals of advanced age, so elderly individuals, and however you define elderly, but it's defined in this case as individuals over 60 years old, which is not to say that younger people aren't affected, and they are. It's just that the higher percentage of serious complications occur in these individuals of advanced age. What is interesting, and we're learning, is that other clinical situations like hypertension, so people with high blood pressure, obese individuals, and individuals with type 2 diabetes. So studies have come out from hospitals that are treating a lot of these patients, both in the United States and Europe and in China. Those populations that do poorly, have more serious symptoms, fall into the highest categories are people that are obese, people that have hypertension, people that have diabetes, and people of advanced age. And so the critical questions that as scientists and clinicians we ask is who, what, where, when, why. So who, that's these populations, why and how. And if we understand the why and the how, we can start to specifically develop treatments to prevent or treatments to eradicate the virus. So can you go to the next slide, Michelle? And these are, I'm just going to show two slides, and these are taken from a paper that was published about a month ago, looking across the board. What we want to know, what we really want to focus on, is the patients who don't do well. So in those categories that I just talked about, and even that can be broken down as the patients that are sick enough to require hospitalization, and then what we learn about the course of that hospitalization in the patients that are successfully treated. So that's what this slide is showing. So the symptoms, which are shown on the left, they have fever and cough, which is what they come into the hospital with, and they have detectable virus. But then over the course of that hospital stay, kind of day 10 or 12 or 14, when a typical infection like influenza, people would be getting better, these patients start to do worse. And it informs us as to why that is happening. And one of these, and Dr. Rada will talk about what we know about the balance of the immune response in these individuals. They're trying to control the virus, but by day 10 or 12, they're losing that battle. And in fact, they're not able to turn down the immune response, and it starts to become detrimental. And so you see in green, that's when these individuals require intensive care therapy. And in purple, the systemic corticosteroids, those are drugs to actually quiet the immune system, because the immune system is out of control in these patients. That's what we've learned. The next slide is the group of patients that we're most concerned about, and those are hospitalized patients that do not survive. And so if we look at the course of their hospital stay, again, late in the infection, by day 12 or 14, things really start to go badly for them, and they require intensive care treatment, including ventilation. We've heard a lot about ventilation, and that is because the damage that is being done to their lungs is enough that they can't breathe on their own. And when that happens, and why that happens, and how the immune system is actually contributing to that, that's the subject of intense study and investigation. And so that we can try and intervene in a very specific way. One of the ways that we know now that we're able to do that is this virus, the SARS-CoV-2, actually infects different animals that we can use as models for this infection. And we see experimentally the same course of infection in some of these animal models. And that becomes very useful because we can understand the process over time in what goes well in some situations and what goes poorly. And taking that information, we can inform the development of therapeutics, drugs, or we can better define what we need for a vaccine to be successful. So that's the kind of research that, that my lab is doing, and others all over the world have mobilized to try and study this infection. And as Michelle said, my lab prior to this, 3 months ago, was working on respiratory pulmonary infections in these kinds of populations, type 2 diabetics, individuals with obesity, and individuals that are otherwise immunocompromised, like elderly patients. And so we try and understand the immune response in those animal models to influenza or other respiratory infections. And now we're applying that very quickly to the COVID situation. So
Karen, can I ask a question? So what is the commonality in these particular patient populations that are having or experiencing the more severe symptoms? Is there a hypothesis, guess?
Yeah, that's a very good question, and we are learning a lot. And one thing that we know is common to these patient populations: diabetes, obesity, aging. Those are all diseases that involve a chronic or a long-term inflammatory process. So there's kind of, our immune system induces an inflammatory response in times of stress, or if there's an infection, but there has to be a control to that. And in those patient populations, we know that for various reasons, there's kind of a slow burn inflammatory response going on all the time. And that's not good. That's not helpful. When you overlay that with a virus like flu, or now we know the SARS-CoV-2, we know that that accelerates the inflammatory response. And in those patients, they're already cooking that inflammatory response at a low level. And when you add viruses like this, it adds fuel to that fire. And it becomes difficult to regulate it and turn it off and turn it down. That's why 10 or 12 days into the infection, when things should be quieting down, they're not. And that these are the patients where that becomes the biggest problem. Balazs, Dr. Rada is going to talk a little bit about that balance, which is so important.
Great, thank you. So to the attendees, I invite you all, if you have questions, to use the Q&A option in your Zoom, and we will do our best to address questions for each of the sections, and those that we don't get to, we will do our best to address those at the end of the chat before we sign off. So that's a great segue into Balazs Rada. He's going to talk to you about kind of the two arms of the immune system, of your immune system, and when they turn on and when they should turn off. And so just as an intro, Balazs is an associate professor in the Department of Infectious Diseases. His general expertise is in what's called innate, or the inflammatory side of your immune system. And he has a special interest in what role this immune system or this portion of your immune system plays in lung diseases. He is currently doing research on cystic fibrosis, the inflammation in the airway of that particular disease, and also other bacterial infections that target the lung. And he's going to provide, like I said, an overview of how your immune system, the two major components, actually turns on and turns off in response to viruses like SARS-CoV-2.
Thank you very much, Michelle, for the introduction. And after Karen's great introduction, I will try to show you the two major branches of the immune system, the immune response, and how we think they are not functioning well in the case of COVID-19. So classically, two major arms of the immune system have been considered, and this view still holds on pretty well. And these are the innate immune system and the adaptive immune system. As you can see in the upper right corner of the slide, the major function of the innate immune system is to trigger inflammation. It's a very early reaction of the immune cells to any intruders, including viral infections in the lung, to make sure that the body responds really fast to this intrusion. It comes with some host damage, which is good, and it's also required to actually ensure that the second branch of the immune system, which is called the adaptive or acquired, has time enough to develop. In this case, the adaptive immune response purpose is to develop so-called antibodies. And two of my colleagues will talk about them later on, so I will focus on the early part of the immune response, which is the innate. In case of airborne pathogens in the lung, the immune response, the innate immune response starts with the epithelial cells. As last week's Fireside Chat already mentioned it, airway epithelium is the primary target for any viral infection in the lungs, including SARS-CoV-2. The airway epithelium is really important in maintaining the structure of the isolation of the host tissues from the outside environment, and of course also initiating the response to these. Certain molecules on these epithelial cells called receptors bind SARS-CoV-2. AC2 was mentioned in last week's talk, and this molecular interaction is really important, that the virus actually enters the epithelial cells, starts its proliferation process, and starts the infection. What the airway epithelium does, they function partially as immune cells, so they can actually produce antimicrobial molecules. They produce mucus, which helps to clear the airways. And on top of this, they can actually release cytokines, which are messenger molecules of the immune system, which helps the recruitment of professional immune cells, which can actually come from the blood, migrate to the site of the infection, and deal with the intruders. So as you can see, they are really important in alarming the entire immune response. Cytokines, as you can see, they are molecules which are needed for a healthy immune response. As the panel A on this figure shows, which I thought is really nicely described what's going on with an acute controlled viral infection in the airways. The red curve shows you the innate immune response, the blue curve shows you the adaptive immune response, and the yellow greenish area shows you the presence of the virus, the so-called viral load, how many viruses are present in the lung. And as you can see, the early innate immune response is the primary peak in the immune response. It peaks. The adaptive immune response is delayed compared to that. And if things go well, they are ready to control the infection. So once the viral load starts to decrease, you no longer need any of these branches of the immune response. So you need to downregulate them. You need natural breaks of this process. the innate immune response typically has to wind down completely by the end of the microbial clearance. The adaptive immune response maintains in forms of memory cells, which again will be talked about later. It was really important to talk about how things go normally in order to understand what's going on with chronic viral infections, which also includes SARS-CoV-2 infection or COVID-19, which is the disease caused by this virus. The B panel shows this really nicely, that what is the problem in general in chronic viral infections, but also in this case, in the case of COVID-19. One of the differences between the two graphs you can see is that the red curve doesn't wind down. So the innate immune response, which is supposed to be controlled early on, in the case of SARS-CoV-2, is not controlled, it's maintained, the innate activation persists. And we think this overactivation or exuberant activation of the innate immune response is really important in the mechanism of COVID-19, and it is a major contributor for lung disease and also the multi-organ failure, what is seen in patients who get hospitalized and also who get really sick. The other problem is, which we just learn more and more about this, that the adaptive part, the blue curve, is not as high as it should be, and it winds down earlier. So definitely it seems like there are problems also with the adaptive immune response, which are also affected by SARS-CoV-2. And as the effect of both the innate and the adaptive immune branches of the immune system, the viral persists longer. So it doesn't disappear as in panel A. The yellowish curve is maintained, persists much longer. So that will turn the entire infection process, which should be acute or fast, into chronic or long-lasting infection. That will lead to the biggest clinical problems. And another term which you can also hear in the news and definitely relates to this introduction is the term called cytokine storm. As I told you, cytokines are really essential players of the normal immune response. Without cytokines, organisms wouldn't even exist. So they are really essential. But now we learn more and more that in case of chronic infections, but also in the case of SARS-CoV-2, cytokines are being produced on a larger level, and they are present for much longer time as they needed, which we call cytokine storm, and they are pretty much become bad things that contribute to the disease in a large way. So the cytokine storm is just an overactivation of mainly the innate immune response, the inflammatory response of your body to this virus, which at the later stages of the disease are definitely considered to be disadvantageous. Could we jump on the next slide?
So, Balázs, can I ask a question? Do we know why the innate or the inflammatory response is staying on or is coming back on at like 10 days, 14 days into seeing the virus?
I would say it's not clear yet. Definitely there are ideas and hypotheses about that. One hypothesis, which is definitely most likely is really strong, seems to be what Karen mentioned, that patients who have already certain underlying inflammatory conditions, in their case, the virus is just amplifies the entire process. So that could be definitely patients who have already underlying inflammation going on. In their case, that's a risk factor. But overall, it's still there are several questions and we are just learning as we study this virus and we try to treat it. It seems like also the virus has a evasion of the early detection by the innate immune system. So the latency, for instance, is much longer than most of the respiratory infections, including influenza. So that's the number one failure when the innate immune system fails to clear it or fails to alarm the body. It's unusually long, 10 days, even 2 weeks latency times. It's a sign for us to say that the virus has some mechanisms to suppress the early innate immune response. Again, these are the signs, the molecular mechanisms. I think they are far from being completely understood for this.
And is it possible that there's a component of the virus that is turning off the molecules that would tone down that innate system? So there are molecules or cytokines that are inflammatory, but then there are molecules that come in and say, okay, everybody stand down, we got the virus under control. So do we know if those molecules that are turning down the immune system, if they're just not coming in, if they're being prevented from turning on by the virus?
So in the case of SARS-CoV-2, I think it's still relatively unknown, but it's for several other viral infections, they are known that the microbes, including viruses, use generally mechanisms to shut down specifically certain branches of the innate immune system or the adaptive. So these are known that the viral, and these are viral molecules, genes, or they target certain normally efficient ways of the immune system to shut them down, to inhibit it, and we call this immunosuppression, and they help the survival of the virus or the viral clearance. In the case of SARS-CoV-2, again, I think it's largely ununderstood.
Okay, thank you.
So the next slide I thought was a very good summary for us to understand. Again, it's a recent relatively recent publication, which shows the relation of the infectious process or the immune response to the directly to the infection versus the exuberant host inflammatory response, and how these two are responsible for the overall disease severity. So as you can see in this curve, on the x-axis you have the time, the y-axis shows you the severity of illness, and the black curve shows you just kind of an optimal or idealized increase in the severity of the illness, meaning that patients are getting sick over time. And this staging proposed by this work has been actually well accepted on a clinical field dealing with COVID-19 in terms of splitting the whole entire disease process into three stages, three clinical stages, 1, 2, 3, as you can see. The figure really nicely explains that the stage 1 is, which is considered the early infection phase, when the immune system mainly directly responds to the virus. And this is when people, this is also happening in everyone who encountered the virus, and typically these are the signs of mild common cold-like symptoms. You might develop fever, dry cough, some headaches, but these are the symptoms which, again, can be directly to the viral response by the immune system. If the virus is not cleared and pertains in the lungs, then the disease gets to its stage 2, which is the pulmonary phase. So the upper airways failed mainly through the innate immune mechanisms of clearing the virus. The virus had a chance to get lower in the lung and starts to infect a few lobes or maybe even all the way to 5 lobes of the lungs in patients. And here, the direct response to the virus is decreasing, while the host inflammatory response, which is this exuberant, unnecessarily uncontrolled inflammatory response, is increasing. And you have a mix of the two. And if the disease develops further and comes to its latest stage, which is stage 3 or the hyperinflammatory phase, is when we think that the virus is not anymore direct. So the immune system doesn't directly respond to the virus anymore, but most of the immune response is directly related to this over exuberant inflammation. And these are also important for us to understand what do you need to target in order to help the patients to become better at different stages of the disease. And that also highlights that at the late stages, main targets actually should be placed on the immune system, not on the virus itself anymore. And so the figure also describes at the bottom the clinical symptoms and the signs what Karen was talking about and mentioned. I wanted to focus here mainly on the relation of the two responses of the immune system. And if you could jump on the last slide. Mine.
Yeah, so there's a couple questions that have come through before we move to the acute respiratory distress syndrome. One question has to do with IL-6. If it's part of the cytokine storm, why is the IL-6 inhibitor the only one that has drawn researchers' attention in treatment? So I'm going to open that up to the panelists. I'll point out that the interplay of cytokines and the various immune cells is complicated, to say the least. And there are certain cytokines that attract other cytokines. But I'll open up this IL-6 question because I'm sure people have heard that IL-6 inhibitors are being used at certain points in the disease progression to help patients.
I can touch on it and then everybody can jump in. That class of drugs is being talked about and tried first, partly because IL-6 is one of the common players that always shows up in these detrimental inflammatory diseases. The other is that it's a drug that we already have on the shelf. So that's one of the reasons why that's getting a lot of attention. There are other aspects and other cytokines and other targets that other people and other laboratories are looking at. And again, what's really important is we need to understand the timing of this inflammatory response. So these cytokines don't all come up at once, and sometimes by the time we see things like IL-6 or some of these others, it's too late. The damage is already done. And so it becomes very important to understand the progression, this time course that Balazs is showing. And again, if we can mimic that in an animal model, we can say, what are the earliest signals that come up that tell us this is going to go badly? And those are the ones that we want to target. Sometimes if we are targeting these molecules that come up later, after this amplification of the response is already in progress, it's too late. So there are many of these targets, some of which we already have drugs for, and people are doing clinical trials with these other ones. But IL-6 is a nice target because we have a lot of information from already clinical practice and use with that inhibitor. Yeah.
Okay. And then another question that's come up is, if the inadequate immune system is a factor, why are children that are usually considered undeveloped as far as their immune system not largely affected? I'll also open that up to the panel as well.
That's a fantastic question. And that's a question that we're all very interested in. And it's a multi-component thing. So if you look at it the other way, the glass half empty, the populations that it's apparent that are very much seriously affected, older people, people with this inflammatory response, that's kind of easier to wrap your brain around. It's harder to wrap your brain around why little kids are not as susceptible to serious consequences. And sometimes and this is my thinking about this late at night, is sometimes an immune response, an exuberant and really strong immune response, is not the best thing. And sometimes if it's a mild infection that they've not seen before, and they can handle it. So when we really talk about the immaturity of the immune system in children and pediatric population, it's really in the very young, in infants and toddlers. But one of the thoughts that I've had is this SARS-CoV-2 virus is actually a part of a bigger family of coronaviruses, one of which is a virus that causes a common cold, and we get that over and over and over again. And Dr. Joyner is going to talk about that concept of memory. If you see a virus one time, you have a little bit of a memory, so the next time you see it, you do better and better and better. Little kids may not have seen as many cold viruses as we have, and don't have that kind of a exuberant memory response. And I do question whether or not older people are more susceptible because they do have that accumulated memory that might affect SARS-CoV-2, and that exuberant inflammatory response may actually be detrimental. That's my hypothesis. So no one has the data for that, but that's a very good question that we're all intrigued by. Chet, do you want to chat about that?
No, I think that was a perfect way to describe it. And I do agree that I think there's many, as you said, Karen, very nicely, it's a multifaceted issue. Kids breathe differently than we do. There's all of that type of physiology that also changes as you age. And that affects outcomes potentially as well. So me and you, as we age, we don't breathe as well. We develop these other complications. Whereas kids, all they're doing is running around and playing. And so that's one component. And in terms of, I think a lot about pediatric patients and their immune systems, because as if you were here last week, and if not, you'll learn, is I think about malaria a lot. And that's really a big emphasis in my lab. And most of those patients are pediatric patients. So they're under the age of 5. And so, as Karen pointed out, the immune system is undeveloped in some ways because it is it hasn't seen as much. It hasn't had the time to get this quote unquote memory so that when it encounters things again, it can deal with it more effectively and faster. But at the same time, the immune system of a kid is also a bit more plastic. So it can move in different directions that once you reach a certain age, you've kind of capped out. And Kim may get into, or can talk a little bit about that, because T cells are one arm of that adaptive response that Balazs is talking or mentioning. They're not coming out anymore after you go through puberty. So there's kind of this, multifaceted things that ultimately are feeding into this that may end up saying, maybe when you have and you just are relying on memory, you might be in a little bit more trouble as well because you're not able to make a new one. And then if you can't make the new one, maybe the memory, as Karen pointed out, gets in the way and causes problems. So I think that there's kind of this, the immune system of kids are in some ways undeveloped, but I think that they are also a bit more flexible. And so maybe that helps them actually do better.
So if I could also say that in addition to this complexity, and most likely the innate immune response is more just natural in kids, and there's no obviously underlying inflammation which affects all the high-risk populations on COVID-19. But there are actually viruses which affect kids or which affect kids way more than adults. So not only their immune system is different, but their immune system's response to different pathogens is different than how adults respond. So it's a really complex issue, and I think it's still far from complete understanding.
Kim, anything to add?
Well, it was something I didn't want to jump ahead to because I'll talk about it when I get to my section because I want to bring it in the context of T cells. So I will loop back around, I promise.
You're muted, Michelle.
So Balazs, we'll go ahead and move on to talk a little bit about the acute distress syndrome. I'll come back to the question about seasonal allergies after we've had a discussion about what's going on in the lungs.
Okay, I will try to quickly describe because I think it's has been in the news and it's the major symptom which affects all the COVID-19 patients who got hospitalized. I mean, the name of the virus is also coming from the SIRS part of the nomenclature. So acute respiratory distress syndrome, or ARDS, is reaction of the lung. It's a overall inflammatory response in the lung, which shows up in some of the SARS-CoV-2 infected patients. It's not unique to them. ARDS can be triggered by other viral or bacterial infections or other stresses to the lung, but COVID-19 patients who get hospitalized, most of them have this too. And it's a major complication in the whole disease process. And this figure, I thought, really describes it well in 3 or 4 different stages with really nice cartoons to show what happens. In the upper right corner, you just see the you see the structure of the alveoli, which is the single dead-end street of the lung where most of the gas exchange happens in our body. And alveoli are covered with alveolar epithelium, type 1 and type 2 cells as they are indicated. And their major job is to allow oxygen to be taken up and carbon dioxide or CO2 to be released into the lung. And type 2 cells, they produce a molecule called surfactant, which is really important to decrease the surface tension of the epithelium and keep the alveoli open. And again, this pictures how a normal alveoli should function. And each alveoli has surrounding red capillaries which contain red blood cells, and they carry the oxygen and the CO2 to the alveoli or away from it. When you get infected in your lungs, deep in the lungs in the alveoli with viruses, including SARS-CoV-2, then a local inflammatory response develops. And as the previous figures described it, in case of SARS-CoV-2, this is not downregulated, will be maintained and goes through a moderate and severe stages. And these are shown here on the following two pictures. And parts of this are that cytokines are being released by the epithelium, which will recruit immune cells called macrophages. They can release additional cytokines. The endothelium, the cell type which covers the capillaries, will contract, which leads to the blood vessel permeability. The blood vessels will dilate. So all of this will ensure that there's more liquid from the blood going to the space between the endothelium and the epithelium, or later on inside of the alveoli. This fluid accumulation is a major problem because it decreases the surface where the gas exchange, oxygen versus CO2 can happen. And at severe stages, at the lowest cartoon, as you can see, all this process has become more exuberant. Additional white blood cells called neutrophils show up. These cells are part of every chronic inflammatory process. And if they are not being shut down, they contribute to tissue damage. My laboratory is actually studying this cell type in the chronic airway disease in cystic fibrosis patients in collaboration with Emory. So it's a really interesting cell type, but they have been related to really the end stage, the stage 3 in COVID-19 patients. And they destroy the tissues by releasing proteases and reactive oxygen species. So at the end, you will have a protein-rich fluid filling up the alveoli, enabling the oxygen exchange between the lung and the blood. This will lead to oxygen or hypoxia in the blood. Several organs will suffer from hypoxia. The tissue damage will further amplify the inflammatory process. Cytokine actually can lead to the blood and turn into so-called systemic inflammation, which is one step in the process to lead to several other organs being affected. And long-term, what also has been seen in COVID-19 patients who survived but had ARDS, that fibrosis happens. So the scar tissue develops in the parts of their lungs, and they lose, many times it's over, they permanently lose, or they have a really long way to recover certain parts of the lung in terms of being able to use it for the function of gas exchange. So again, this is the description of the ARDS, which has been seen in most of the hospitalized SARS-CoV-2 infected patients. And this shows you just, again, how the innate immune system and exuberant inflammation can actually help in the destruction of your own tissues.
And so, Balazs, we know from various reports that clinicians are actually looking for increases in neutrophils to help stage patients and understand, what therapies to put them on next. So it seems like it's not normal to have an influx of neutrophils into the lung. Is that correct?
So I wouldn't say it's correct. Temporarily, you can have them. They are typically should come in, clear the infection, and they should be stopped from coming in. The problem is the chronic part of the entire process. because they stick around for longer, and obviously they don't help completely to remove the virus. The virus persists throughout this whole time. So again, it's not a direct response to the virus anymore. It's a hyperinflammatory stage. You have too many of them for too long, and that's the main problem. And sooner or later, they will release the dangerous antimicrobial cargo, which in every case, I can list you 20 different diseases when neutrophil-mediated chronic inflammation is a major problem. And they are one of the leading to tissue damage in the clinical signs. So therapies, to my knowledge, to target them, they're still pretty rare, but definitely at the end stage, they should be targeted because high neutrophil numbers or neutrophil lymphocyte ratio has been clearly linked to severity in COVID-19 patients.
Okay, so a couple questions along those lines. Is it possible that inflammation caused by seasonal allergies is exacerbating the symptoms of the virus? Have we seen any evidence of that to date? And I'll open that up. Balazs, if you want to start, if there's anything else from the other panelists.
I might not be completely updated on the literature. I've seen links to asthma, which is a more severe autoimmune form of hypersensitivity. For sure, so with asthma, again, I thought the respiratory immune system is kind of, again, primed, ready to jump on anything, and viral infections others than SARS-CoV-2 have been shown to exacerbate it. But I'm not sure about studies on seasonal allergies. I don't know if anyone else has some literature to cite.
I mean, allergies is primarily a mast cell mediated disease, and it's kind of skewed less away from this inflammatory response. So actually in a lot of ways, you would think it would kind of be a better thing, not a better thing. I mean, it's not going to help you with this virus, but it's certainly not going to push that inflammation that we're seeing with the infection. Now, definitely very different with asthmatics. I mean, I'm just talking about the standard seasonal allergy. Asthmatics are in a totally different situation because a lot of those patients have had significant airway remodeling and other, changes in the physiology of their lung and their immune response that probably does put them at a greater risk.
They also, as we all know, take a lot of different drugs, and a lot of the very effective things to deal with some of this remodeling and stuff that Kim talked about are some of these drugs that modulate the immune system in some way, like Dr. Norris said. So the IL-6, for example, is one example of that, but there are others that are used for allergy patients. And we actually have no idea, does that contribute to some of the things that we may see? And, really, as Dr. Norris said really nicely, how do you have to start using your animal models to ask these questions very carefully? Because just someone, is asthma really the underlying issue, or is it the medication someone who has asthma is on that's the underlying issue?
Yeah,
So
at least at the beginning, which, we're only 3 or 4 months into this, but when in studies that have looked at thousands of hospitalized patients, there was a study out of New York City and just looked at all the patients, said who did well and who did not do well and so on. And, over 90% of them had some other underlying issue, obesity, aging, and so on. And they really quantified that and said, what are the top ones? And what was curiously pretty far down on the list was asthma, because we would all think that asthma. So the question was, where are all the asthmatics? And I think it's partly what Kim is touching on is inflammation is not just one flavor. There are variations on it, and inflammation that causes allergies and asthma is one type and has different molecules, and the other that causes this anti-infective inflammatory response, that's a little bit different. And it is curious. We all expected the numbers of people with allergies and asthma to sort of flood the system. And so far that doesn't seem to be the case. So that tells us a little bit, that gives us another clue as to what inflammatory responses are detrimental. Not to say that asthmatics are not affected by this, but the real top players in this are the types of inflammatory response that we know are associated with obesity, diabetes, and hypertension. So we're still putting all the pieces of the puzzle together, but yeah, it is curious that type of seasonal flu or seasonal allergy and what then progresses to asthma, that seems to be different. It's a different patient population. Yeah, that's interesting.
Okay, and then one last question that was posed. How does ARDS differ from bacterial pneumonia or secondary kind of lung fungal infections that patients on the respirators are experiencing? So I don't know if you want to start, Dr. Rada, or if you want to kind of punt it over to Dr. Norris.
So if I can just add, ARDS again, it's not specific to SARS-CoV-2. It's a hyperinflammatory response of the airways, and several stresses can induce it. There can be microbial, so pneumonia, but whatever causes pneumonia can trigger it. It could be viral, bacterial, fungal infection, and the response, or ARDS, the symptoms are relatively similar. It can be a trauma to the lungs, other type of stress, radiation. So a wide variety of stressors can induce ARDS. So they are fairly similar. What seems to be different, that still there are some minor looking differences with COVID-19 ARDS, which physicians just started to realize. This is called high compliance ARDS versus typically the low compliance ARDS, which has been seen before. So they are starting to pay attention that they are different from ARDSs depending on the microbes, which it's mainly important, I think, at this stage for the physicians and nurses working in the patient care, which might be different from the typical form according to the original ARDS definition. But overall, this is again relatively non-specific response of the lung to several stressors.
Sorry, I'm having mute-unmute issues. What about what's going on with patients that are developing these secondary infections after being on ventilators? My understanding is these are pretty distinct from ARDS. Do you want to address that?
Yes, secondary infections have been seen in patients, and they can contribute to the ARDS symptoms, but again, it could be different from the primary cause, which is the viral infection.
Yeah, I think if I could just say
okay.
quickly, the secondary infections are being seen in about a third to half of the patients, the hospitalized patients, in that time frame, at 10 days to 2 weeks, especially when they require ventilation. And that compounds the problem because that amplifies the inflammatory source, because now the body is trying to deal with that too, and it just overwhelms it. And that's why these become such devastating clinical situations for these longer term hospitalized patients once they get on the ventilator. And that is associated with a poor outcome because they're dealing with either a second bacterial infection in the lung or fungal infection in the lung on top of the virus. That's why this is so clinically devastating in these patient populations. Mm-hmm.
Okay, great. So now we'll move on to a second component of the innate immune system centered around certain T cell lymphocytes. And Dr. Kim Klinowski is going to kind of walk us through the importance of this component of the immune system. Dr. Klinowski is an associate professor in the Department of Cell Biology. The major research focus of her lab is understanding how the respiratory environment uniquely programs virus-specific T cells. She has a concentration on trying to understand the development of memory with these T cells, and she focuses on various areas of respiratory diseases as well as traumatic brain injury and immune and nervous system interfaces that affect injury outcome. So Kim, I'll let you go ahead and get started.
Yeah, thanks, Michelle.
Just one we're little time check. We're at 1:55. We've typically been going about 90 minutes, but I will do my best to definitely get us through and keep you at the 90-minute, if not sooner time frame. Thanks, Kim.
Thanks, Michelle. And I appreciate the opportunity to kind of introduce this cast of characters to the audience today. So all of the cells, these cartoon cells that you see here, are lymphocytes. But the ones that I'm going to focus on today are the ones that Michelle has highlighted here, and these are T cells. Dr. Joyner will talk a little bit later about B cells. So T cells, there's 3 different kinds that you see here, and they're all very functionally different populations, but they really work together collectively to protect us essentially from a wide range of infections, both bacterial as well as viral. And they also, especially that police officer regulatory T cell, we won't talk too much about him today, but he is kind of the balance of these other 2 populations of T cells. And this population of T lymphocytes is important for inadvertently causing us to cause damage to our cells. So I'm really going to focus on that first population of cells that you see there. Those are the CTLs, and those are CD8 T cells. They're also called cytotoxic T lymphocytes or killer cells. And you can see the one that's shown here is in the form of a ninja. And these cells are important because what they do is they actually kill host cells or your cells that are actually infected with the virus. And in this way, the virus can no longer essentially replicate inside our cells. And then, of course, the reservoir of the virus can be eliminated. So we have individual T cells that recognize very specific viruses. So the ones that you see here are very specific for SARS-CoV-2. So these would be, that's supposed to be portrayed to you because of the fact that they have the little viruses on their shirts. So each of you have different populations of these CTLs that very specifically recognize these individual viruses. So what happens is when there is inflammation, so we've talked about this kind of activation of the kind of inflamed cytokines and things, they activate these T cells along with the presence of virus in a way that they then replicate themselves. And that's what I was trying to demonstrate to you here, is that you now have an army of these cells that will then go out into your tissues and again, hunt down that virus and really remove that reservoir of virus. So in a perfect world, let me just kind of tie you back around to what some of the things that Balazs talked about so you can kind of understand where this population of cells really fits into this entire response. So where we started our discussion today was on that innate immune side. So that is, if you look at that top graph in A, it's to the left of the dotted line. So you can see there's some cytokines. An example shown here is type 1 interferon. Some of those innate immune cells, that's the purple line. So again, they're kind of coming into importance in that early response. And what they're doing, or what they would do in a perfect world, is they would keep the level of virus in your body down until those cytotoxic T cells have time to make those copies of themselves. So if those cells weren't there, that virus titer would kind of keep going. So essentially, I think everyone's kind of familiar with this concept of flattening the curve. It's these innate immune cells are essentially trying to flatten that virus curve for a little bit of time so that the adaptive immune system can become activated and really control the virus. And that's important, and I think you can appreciate that these virus-specific CTLs are really important in this response, because you can see as they increase in number over time after infection, the actual virus titer will go completely down. So you can see as that red line goes up, that green viral titer starts to go down. And so these cells are really important, and in particular in a naive population, so a population such as we are now that has never seen this virus before. Yes, who has never seen this virus before, because this population is really key to elimination when we don't have a good pre-existing antibody population or a B cell population in our bodies. So the thing about T cells is they do cause a little bit of collateral damage. So there will be some death of those cells, and that's why antibodies are always the preferred immune response. So I just want to kind of put this all together for you in kind of one big perspective, so you can kind of understand how the adaptive immune system responds during a normal infection, and then we can kind of talk about what happens and what goes awry in response to SARS-CoV-2. So if you can kind of imagine that your body is a city, normally your body is not on fire with inflammation, but in this particular analogy, what I'm trying to depict here is that there's some type of inflammation. So whenever you get infected with pretty much any pathogen, so any virus or any bacteria, you get this initiation of inflammation. But what will happen is, because there's this kind of specific signal that's given by this particular virus, those T cells that I talked about, those ninja cells that are specific for SARS-CoV-2 will become activated. And again, those cells are going to go out into the tissues, and then they're going to attempt to clear the infection. But what I want to talk about for a second here is that these cells, these CTLs, are kind of only one part of the equation. So this is kind of the entire immune response from these CD8 T cells over time. And what I want to point out is that while that army is really important for clearing the virus, there's also another population, a subpopulation of these cells that will develop later on. And you've heard us throw around this term memory. So these are the cells that are going to stay around for a longer period of time. They're going to become quiescent, so they're not going to be just going around and killing cells at random, because at that point, when they form, the virus is actually gone. So the virus has been cleared. So these memory cells are really important because you can see they stay around at a higher number than they were present before infection. But if another virus, that identical virus, comes back again, they can become reactivated quite quickly. So they're really important for re-challenge. So when you get infected again. And my lab is very interested in that particular population of cells, because again, they can provide this kind of long-lived immunity to this particular virus. So just to kind of tie this into the context of what we're seeing with this particular virus, is if I can focus your attention on the bottom part of the seesaw where everything is balanced. This is a good example of what happens during that transition where cells go from effector to memory T cells. So what happens is, as those ninja cells are out in the body and they're clearing the infection, the actual numbers of viruses going down, and because that is going down, you also tend to have a reduction in that inflammation that we talked about. And then in addition to that, you also have that suppressive population, that COP, who's kind of also regulating this response and making sure that the CTL response is not essentially going out of control. So once all of that happens and the virus is essentially cleared, inflammation is gone, what happens is you get development of those memory cells, and the system kind of returns to that balanced state. Now, what we're seeing, or what studies have been showing us, is a couple different things with this particular virus. So because you have this very high level of inflammation, what's happening is it's not clearing. So those ninjas, they're out there and they keep trying. They're just trying to kind of fight this fire. And as they're doing this, what can happen is you can eventually develop what's referred to as immunopathology. So that's when your immune system is actually causing the pathology that's non-specific or shouldn't happen under normal circumstances. And of course, that can result in tissue damage. Now, the other thing that can happen, and Balazs alluded to this earlier in his discussion, is as these cells, as these CTLs are out and they're trying their best against this high level of inflammation and viral replication, they might fight so hard that they eventually become what's referred to as exhausted. So they lose their ability to really do anything. So they become essentially non-functional. So all of this prevents the development of memory as well. So my lab is really interested in the research that we're working on is what are those signals that are really important in the lung environment that are essentially driving the development of T cells and their development into memory T cells as well. So that's an important area of research that we're working on. And I just kind of wanted to end by I've kind of ignored this CD4 T helper cell over here on the right. This is another important lymphocyte that I think Chet will talk about a little bit in his discussion. These are cells that you may have heard of that are referred to as helper T cells. So they both help CD8 T cells, but then they also help B cells in making a really good antibody response. So that was my presentation that I had to kind of introduce you today to T cells and kind of their very important role, not only in responses to viruses in general, but particularly in response to this virus that we as a population have not seen before.
Thanks, Kim. So questions come in about in relation to flu. So I'll ask two questions together. One is, I'm assuming memory T cells are going to be important for good response to any vaccine. And the second question that has come in is, why doesn't our body create these memory T cells for influenza?
So that is actually one of the big focuses of the research in my lab. That was a perfect segue. So it's really interesting. You can generate really strong, long-lived T cell memory in responses to a lot of infections that are coming in from various sites, places like the skin and the gut. They tend to be well suited to kind of support these cells for a very long period of time. And I failed to mention this in my talk, but a lot of these T cells are actually in the tissues. They're in the sites where the infection initiated. But we don't see long-lived memory cells that stay in the lung for a prolonged period of time. And we think, because of the fact that the lung is such a vital organ, if we had lots of memory T cells that accumulated over time for all of the respiratory infections that we essentially encounter, we might not even be able to breathe, right? So there needs to be, some regulation in that site. So we know that you can generate short-lived memory T cells that will go into the lung, but there's been studies for decades that have shown that there is just, they're just not supported for a very long time. And the reason why, my lab and other labs are actually working on that. And I can't remember what the other question was. Something about vaccines?
The other question was, how important are generating memory T cells? How important is that going to be for developing a vaccine against SARS-CoV-2?
Yeah, so the approach and Chet will talk about this for sure, the approach that most of the vaccines that are being developed for SARS-CoV-2 are antibody-based. So they're trying to really elicit a strong antibody response. And I alluded to this in my talk, is that, T cells are great. They will certainly clear your body of a virus, but that's not without getting sick. Because T cells are recognizing the virus after they've gotten into your cells. So we typically try to generate vaccines that are very antibody centric, because if you can develop a good antibody vaccine, you can essentially prevent those viruses from even entering the cell and causing disease. So that's typically the gold standard for vaccines. But that's not to say T cells aren't important. And again, I think Chet's going to hit on this, is that those helper T cells are very important. So we need to make sure we activate a really good CD4 helper T cell response, because they are so intimately involved in a good B cell response, so a good antibody response. So I think he's going to talk about that, but this is But I don't want to say that CD8 T cells are not important, because the other thing that they do that's a little bit more beneficial in terms of B cells is they can actually, they tend to recognize parts of pathogens that tend to be conserved between different strains. So a lot of T cells, so the T cells that we study, recognize in influenza a part of the virus that is found in lots of different strains of influenza. So things that aren't mutated or changed to avoid that antibody response. So there is, for some diseases, for some viruses like HIV, like influenza, some parasitic diseases like malaria, ones that are highly mutagenic, those T cell based vaccines are becoming more of a target for those types of infections. But things that aren't as mutagenic, and we have no indication, at least yet, that this virus is going to be heavily mutagenic, you would be really pushing towards a B cell based vaccine.
Great, thank you. So with that, we will move to Dr. Chet Joyner, who's back with us to talk about antibody responses to this virus. So just to refresh people, Chet Joyner is an assistant professor in the Department of Infectious Diseases. His research is primarily focused on understanding the role of antibodies, B cells, and plasma cells during infections and vaccination. His work is focused on antibody responses, mostly, like he said earlier, against malaria parasites and how they protect against disease and transmission. But he's also interested in understanding the role that antibodies are playing with respect to infection of the SARS-CoV-2, both singly and potentially co-infections with other diseases. So with that, I'll let Chet take it away.
Thanks, Michelle. Thank you for having me back to talk a bit more about antibodies in terms of their kind of basic biology and how they work, versus last time I was here, we talked a lot more about how we apply our knowledge of antibodies, for example, to develop a diagnostic. But I think the way to kick us off is a little bit of redundancy and just ask, what is an antibody, and refresh the audience and everybody's minds about what antibodies actually are. And these are the proteins that are produced by cells of your immune system in response to some foreign molecule that your body views as not being itself. So in our case, talking today, we're going to be thinking about it in the context of an infectious microorganism like a virus, a bacteria, a parasite. And really, these antibodies are made to neutralize that foreign entity and get rid of it outside of your body. They're beautiful molecules, as you can see from the 3D structure on the bottom left-hand side of the slide. And they're very complicated, and made up of multiple pieces. But ultimately, the goal is to make the parts at the end that you can see that I've highlighted in green, enable them to interact with, for example, a virus. So if you imagine this is a virus, your antibody's tuned to fit that virus and hold on to it and, for example, stop it from invading those epithelial cells and creating that damage and preventing that from happening that everyone's talked about over and over today. And by doing that, you also tone down that inflammation and all those things that Dr. Norris talked about as being bad. To have that for a long term of time is not a good thing. There's many types of antibodies, which we talked about last week. Some like IgG, which you're also seeing on the left-hand side here in the structure, is really a workhorse and is probably being studied a lot in terms of identifying types of antibodies that neutralize the SARS-CoV-2 virus. That's not to say that other types of antibodies aren't important. For sure, IgM antibodies, which are produced early, are also very important in controlling the virus. And maybe even IgA antibodies, which are typically found, for example, in your mucosal sites, like lining your nasal cavity, in your lungs, in your gut tract. These may also be important. So there's a lot of active research happening at UGA and across the globe, and asking what antibodies are important and how do they actually work to control the virus and eventually clear it from your body so that you don't have the sustained immune response and have a bad outcome to the infection. So next slide, Michelle. So first, let's just focus for today, though, on IgG and IgM. And let's just ask the simple question of when are they made? And I think that's important in understanding how antibodies evolve over time and why that is important for controlling and clearing, ultimately, an infection. So you're going to get infected, in our case with SARS-CoV-2. And as Kim showed very nicely in her slide, you're going to have a virus start to replicate, and then that innate immune system is going to kick off. You're going to get some T cells starting to get activated. And in my case, I'm interested in antibodies, and we're also going to have cells get activated that we'll talk about in a second that are going to result in the production of these IgM antibodies. And one of the key functions of these IgM antibodies is to really flatten that curve, as Kim put it, contain that virus while all these other activities of the immune system, whether it be the T cell response, more innate immune responses, whatever, can work together and really bring that virus to a halt and bring it down. Part of that process is these IgM antibodies are going to be produced, and then some of them are going to go on to become these IgG antibodies that are actually better at that fit. So they actually fit that virus a little bit better, and they can ultimately control it and hopefully contribute to the clearance of that viral infection. Now, what I want to point out, can you just go back one more time, Michelle, is that even after you've cleared the virus, IgG sticks around for a long time. So IgG, unlike IgM, which is more associated for when you have the virus around in your body, IgG sticks around for a period of time in your plasma. And for these SARS viruses, it seems at least for about a year, and without you being re-exposed or reinfected. And so I want you to just see that, because we're going to talk about kind of how that occurs and why that occurs, and how these IgG antibodies persist and why that's important in, say, protecting you from reinfection at the end. So next slide, Michelle. So now that we know what antibodies are, when they're made, let's talk about what cells actually result in the production of antibodies. And these are those B cells. So B cells are cells in your body that all of us have that actually have little surface antibody molecules. And they're floating around, surveying throughout the different parts of your body. And when they come in contact with an antigen, which it's just something that causes an immune response. It could be a protein of a virus. It could be pollen, if you have allergies, for example. And these B cells are going to recognize and interact with that antigen and go, wait a second, this isn't supposed to be here. Let's see if we should respond to this. And that's when these activated B cells are going to start talking to their friends of the immune system, for example, those helper T cells that Kim referenced. And those helper T cells and the innate immune cells that produce those cytokines and things are going to tell this B cell, you know what, you should get activated and respond and produce your antibody that you have sitting on your surface. And that happens in a variety of different phases. But as Kim said, that B cell is that's specific for the virus or whatever it's encountered, it's going to rapidly proliferate, make many, many, many different copies of itself, and undergo many different processes. So the first thing it's going to do is some of them are going to go on to make what we call plasma cells. Now, plasma cells look different than a B cell, as you can see here on that very top line where you see the IgM molecule. And the first B cells that get activated, part of them will go and make these plasma cells, and they're going to make those IgM antibodies. And that's that first wave that I talked to you about and mentioned on my slide previous to this. Some of these B cells are going to actually undergo, because of the helper T cells and other signals from maybe even the innate immune system, they're going to go on to become these IgG expressing B cells. And this is through some intricate processes of the immune system we don't have time to get into, but nonetheless, you will get now what we call isotype switching. So this is only a B cell that's decided, you know what, I'm making IgM. It seems to be working pretty good as I talk to my buddies in the immune system. So I'm going to make an even better antibody of clearing this virus known as an IgG. And that's how you get IgG. So that hopefully tells you you start with the M, and then, some of those are really good and they become G. And that's how that process occurs. Now, during that whole phase of going from M to G, so on and so forth, your B cells are even going to further what we call mutate, so that that fit that I talked about becomes even better. So they're actually going to generate even better antibodies that bind to those viruses and hopefully neutralize them even more effectively. And we call that affinity maturation, which is just a fancy term for making sure that that antibody fits that parasite, bacteria, what have you, as best as humanly possible. Now, at the end of all of this, when those antibodies have been made by these plasma cells and we've gone through this kind of elaborate process, hopefully you're left around with what we call some memory B cells. So like Kim said, these are memory cells that persist and hopefully will stay with you for a really long time. We don't know the answer to that about SARS-CoV-2, but that, for example, is something my lab is really interested in asking, that stick around and ultimately give you some sort of protection against reinfection. Regardless if it helps you prevent you from getting as sick as the first time you got sick, or completely protects you from a reinfection entirely. But all of that is really still at the cutting edge of research and up for debate. So if we go to the next slide, Michelle, kind of to drive that home, I think that when you think about antibody responses, T cell responses, you know, we have to ask ourselves, like, why would we want memory to be around? And the way the antibody response protects you from future infections is you go through in your first infection, shown here in blue, that whole cascade of events that we've already talked about. The naive B cell gets activated, you get those short-lived plasma cells making IgM, maybe some making IgG after a period of time. That's going to neutralize the virus. And once that virus is gone, you're actually going to have some plasma cells hopefully stick around and make that IgG I talked about on the second slide. And those are actually what we call long-lived plasma cells. And they're kind of the gold standard for most vaccines. They sit in your bone marrow decades after you initially encountered an infection. You got vaccinated, for example, against yellow fever, a virus that causes disease in the tropics, and you're traveling on your vacation and you have to get that vaccine. And they stay and make antibodies that hopefully protects you against reinfection. You also get these memory B cells that stick around. And the whole purpose of that is that if you get another infection, those memory cells are sitting at the ready to respond even more rapidly, which clears that virus even more effectively because they now have better antibodies that they've made through that whole maturation process we talked about. And they already have IgG. You don't have to go through that whole long-term thing of, well, first we have to make M and then we're going to make G. Nope, you've got the G on the ready and it's really mutated. It fits that virus very well. And hopefully that second infection will actually produce even more long-lived plasma cells that'll keep churning out IgG even after you clear the virus and produce even more memory B cells. And this is also, you know, going into next week's talk, the same exact concepts of generating a vaccine. So applying some of this knowledge, I think there's been a lot of questions out there because of some data that got produced early on in this pandemic from animal models saying, can people become infected again with SARS-CoV-2? We don't know, is the bottom line answer to that. There is some data from animal models that suggest that in the short term, and this is the very short term, about 28 days after something has been cleared, that you're protected against reinfection. But that really may not be the reality of what humans will face, where we have 6 months between our exposures, for example, if this becomes a seasonal virus, as some people speculate it will. So this is one of the questions that my lab, as I said, is very interested in understanding what's the contribution of these memory B cells versus, say, plasma cells. If you get infected again, I think this is important. Will you get as sick as the first time? Again, we just don't know. And this is another question we can answer at UGA with all the people on this panel and others of our colleagues in terms of using our animal models. We also talked about this in the diagnostic section, but I just want to drive it home one more time. Not all antibodies are actually neutralizing against the virus. As I said, if they don't perfect that fit or that interaction, they may stick to that virus, but they may hang on like this. And so that virus is still able to kind of do its thing, but it's not able to actually catch it and say, nope, you can no longer get into this cell to make more copies of yourself. And so even though, for example, you have a positive titer after you go and get your antibody test, it absolutely does not mean that it's neutralizing. And this is something that our colleagues like Dr. Jared Musa and others are actually looking into. What do you need to get a neutralizing antibody? Where does it interact with that virus to stop it? And then finally, just to close, you know, I think the concept of a durable immune response, and for those who haven't heard that, is an immune response that will last a really long time, is we still don't know about that with SARS. And that's, I know I've said it over and over, but I'm hoping you take that SARS-CoV-2, you take that home with you. It looks like some of the other viruses in this family generate long-lived plasma cells, for example. These cells that I'm showing you here in blue that come actually out of bone marrow from patients, you know, we don't know if we get these against the virus. And furthermore, we don't know if they neutralize the virus or what they do. And that is really one of the big questions that my lab and my colleagues really want to answer together is, do we generate these cells and do they last a long time to provide you with confidence as you're going to travel, go back to work, whatever, you know, what's your risk of being reinfected and how do we mitigate that risk and design interventions going back to the very beginning to help those people that do actually get reinfected. And that's it.
Thank you, Chet. That was very informative. And that actually segues us into kind of a snapshot, and this is a very narrow snapshot of what type of research is underway at the university and in the College of Vet Med. So we're certainly trying to understand when does viral shedding begin, when does it end? Like Chet said, identification of antibodies that inhibit the SARS-CoV-2 virus, these so-called neutralizing antibodies. How long are they present after infection? What concentrations of these antibodies confer protection? We're also trying to address the immune response to reinfection, identify predictive markers for those that might have severe outcomes. We're looking at underlying immune response in animal models that mimic the symptoms that we see in people. Also in those patient populations that seem to have some of the more severe outcomes, we're looking to address all of the above questions in these specialized animal models in a very controlled and methodical way, and look at each of the components of the immune system. And I think today you got a snapshot of a very specific, but only a fraction of the components of our immune system that are being utilized to clear this virus. We're also looking at some of the diseases that our hyperactive immune systems are inducing. So the acute respiratory distress syndromes, these hyperimmune responses that are starting to be seen in children, related to, I'm sure you've heard Kawasaki's versus a toxic shock syndrome, the coagulation issues that people are developing that had very minimal respiratory symptoms, but in week 3 are developing blood clots. It's just not clear why our immune systems are reacting the way that they're reacting and when in the time course of the disease that they're reacting. So there's a large amount of research in the various scientific research departments within the college and within the university to try and understand the underlying issues with this virus. And I will segue from that into our Fireside Chat for next week, which we're actually going to talk about vaccine development and various treatments that are available now and treatments that are being developed to target this particular virus. So we will bring back Chet and Karen to talk about vaccines and therapeutics. Jared Musa will join us to talk about antibodies, therapeutic antibodies, so-called neutralizing antibodies. Karen and Biao-He will talk about vaccines that are under development and how vaccines work. And then Jeff Hogan will talk a little bit about the mechanisms of antiviral drugs like remdesivir that you've been hearing about. So with that, I'd like to thank our panelists for joining us today, and thank you all for taking time out of your day. Hopefully this information is useful. And if you have questions, please feel free to email those in to Kelly, and we will try to get responses back to you. Thank you all again. Wash your hands and stay safe.